Hello, my name is Joshua Gabi. I welcome you to Coordination and Control. Questions 51 to 56. Explain phototropism in plants. Part B. What is the significance of tropism towards plant diversity? Explain phototropism in plants. Phototropism is a plant's response to unidirectional light. Shoots being positively phototropic, roots being negatively phototropic. When a plant shoot is subjected to unidirectional source of light, the auxins on the illuminated side absorb more light energy than on the non-illuminated side. Therefore, auxins are translocated to the shade side, and less auxins are transported to the illuminated side. This results in the differential growth, in which the non-illuminated side experiences greater cell elongation than the illuminated side, a result in which is bending of the plant shoot towards the source of light, which we refer to as positive phototropism. Uniformly subjected light to the shoot results in no curvature of the shoot due to uniformly distributed auxins. For a plant root exposed to unidirectional light, auxins concentrate on the non-illuminated side, inhibiting growth on that side, such that the root grows away from light, which we call negative phototropism. Part B. What is the significance of tropism towards plant diversity? Positive phototropic response of the plant enable photosynthetic organs, which are the leaves, to access light so as to effect photosynthesis, and this increases the gross productivity of the primary producers. Positive geo and hydrotropism of the roots ensure access towards water, an important raw material for photosynthesis. Gross productivity of the primary producer is increased. Tendrils are positively hapto stroke thigmotropic, thus trying around other plants for support and to access light. The pollen tube are positively chemotrophic and negatively aerotrophic. They grow towards the ovule to effect fertilization, increasing the reproductive rate of plants. Population rises. Diageotropic response in plants. Parts like the rhizome and the runners, they help to colonization of new areas and to avoid overcrowding. Insectivorous plants, being positively thermotropic, enables them to catch prey on contact. Number 52. Describe the significance of chemical composition, arrangement, and distribution of photoreceptors towards the normal functioning of the mammalian eyes. We shall start with the significance of chemical composition of the mammalian photoreceptors. The rods, these ones contain rhodopsin, which is bleached on being struck by low light intensity. Further bleaching of the trans rhodopsin to opsin and retinine results in depolarization of the rods and production of the generator potential. Which generator potential, if raised to a threshold, evokes action potentials and fire impulses via bipolar neurons and optic nerves for vision in dim light due to rapid decomposition and regeneration. We have the cones. On the other hand, contain iodopsin, which is breached to trans-iodopsin from cis-iodopsin upon being struck by high intensity light. Iodopsin is slowly decomposed and regenerated, and the bleaching depolarizes the membrane of cones. Thereby producing a generator potential which builds up to the threshold. This evokes an action potential and fires impulse via the bipolar neuron to the optic nerve fiber, enabling focus in bright light. the slow decomposition and the regeneration effect of bright light vision. The cones are of basically three forms: the blue, green, and red, whose differential stimulation result in other different colors and shades perception. Arrangement significance of mammalian eye photoreceptors. Rods. They show retinal convergence, in which three rods synapse with a single bipolar neuron. This results in spatial summation and high sensitivity of the rods, since the individual generator potential of all rods are summed up at the synaptic region to cause action potential in bipolar neuron. This increases sensitivity in the dim light. Three rods also associated via lateral neurons or horizontal cells, which effect mutual inhibition on simultaneous stimulation of two receptor rods, which are adjacent. This causes sharpness of what is focused cones. Cones adjacent cells are connected by macrin cells to coordinate their function. Cones do not show any retinal convergence. That is, a single cone synapses with a single bipolar neuron, which increases visual precision or acuity. Let's look at the distribution significance of the mammalian photoreceptors. The rods all over the retina, increasing the chances of receiving light, stimulation, and consequently increasing sensitivity. Cones, they are packed at the fovea, which increases visual acuity. And chances of light from close object falling on two cones, which are at least separated by the cone, hence reducing mutual inhibition. #53. How does the size of the stimuli affect a nerve impulse? Part B. How do the following muscle fibers work? 1. First twitch muscle fiber. 2. Slow twitch muscle fiber. 3. Discuss the major properties of synapse. How does the size of a stimuli affect the nerve impulse? Does not affect the size of the action potential. Action potential is all or nothing. When stimulus intensity or size reaches or exceeds threshold, an action potential is evoked, and that's what we call all. When the stimulus size does not reach the threshold, no action potential is evoked, and that one is referred to as nothing. Larger stimuli increases the frequency or the number of action potential. How does the following muscle fiber work? 1. Fast twitch muscle fibers. Fast twitch, this provides a powerful but short-lasting contraction found in biceps and sprinters, adapted for anaerobic respiration, has thicker myosin for powerful contractions, contains more enzymes for anaerobic respiration. It contains a lot of phosphocreatinine for providing phosphate to ADP to reform ATP, anaerobism. Slow twitch muscle fibers. Slow twitch provide less powerful but long-lasting contractions. They are found in thigh muscles and marathon runners, adapted for aerobic respiration, has high-rich blood supply, contain many mitochondria, contains glycogen, and contain myoglobin, stores oxygen. Part C. Discuss the major properties of synapse. Unidirectionality. Action potential, nerve impulse travel in one direction from pre to postsynaptic membranes. Presynaptic membrane has the neurotransmitter. Postsynaptic membrane has the receptors. Filters out low sensitive stimuli. Low level stimuli do not release enough neurotransmitters. Therefore, not enough sodium ion channels open, and not enough sodium ions enter the postsynaptic neuron for threshold to be reached. Therefore, no action potential produced. Summation. Low-level stimuli add together to release enough neurotransmitter to produce an action potential in the postsynaptic neuron. It can be temporal, low-level stimuli present for extended period of time, or spatial, low-level stimuli from a few presynaptic neurons add up together. Inhibitory. Normal synapses are excitatory. Some are inhibitory, preventing action potential from occurring by making the postsynaptic neuron hyperpolarized. Fatigue stroke adaptation. If synapse is bombarded with impulses at a high frequency, with time, postsynaptic nerve cells cease to respond. They become adapted, and impulses are no longer generated in it due to exhaustion of supply of transmitter substances, and its resynthesis cannot keep pace with the rate at which the impulses reach the synapse, thus fatigued. Photo 54. Explain each of the following as applied to receptor cells. Flicker fusion, adaptation. Part B. Explain what causes vision using the fovea to be in color, to have high visual acuity. Explain each of the following as applied to receptor cells. Flicker vision. Flicker is the shining repetitive stimuli of light falling at a receptor cell at a high frequency. Thus, flicker vision refers to a phenomenon where a receptor cell fuses several stimuli into one continuous stimulus due to high frequency of its stimulation. For a receptor cell to detect several stimuli, they should be at a frequency sufficiently lower than the time taken for the cell to generate the action potential and repolarize before the next stimulus is received. If the frequency of stimulation is increased, there comes a point where the generator potentials fuses, resulting into a continuous stream of action potentials being discharged by the receptor, and now it cannot detect separate stimuli. Adaptation. Explain how each of the following is applied to receptor cells. Adaptation. Adaptation is the failure of the receptor cell to respond to a stimulus as a result of its steady, prolonged stimulation. If a steady stimulus is maintained, the generator potentials gradually decline and the frequency of action potential decreases. With time, generator potentials fall below firing threshold, and no further action potentials are discharged. Thus, the receptor is adapted. It is as a result of receptor membrane becoming less permeable to influx of sodium ions. Part B. Explain what causes vision using the fovea to be in color. Cone cells have photopigment iodopsin in 3 different types, which are sensitive to blue light, green light, and red light, thus can detect different wavelengths of light and their corresponding colors. To have high visual acuity. Visual acuity is ability of the eye to resolve 2 or more close stimuli and perceive them with precision. Cones have no retinal convergence. Each cone cell synapses with a single bipolar neuron, which in turn connects to a single optic nerve, thus able to send separate signal to the brain, allowing precision or high acuity. Cones, they are densely packed at the fovea, located at the center of the visual axis of the eye, which is the best position to receive focused light. Lateral inhibition of 2 adjacent cone cells sharpens the contrast between two close images. #55. Discuss the different reactions of plants to light. Discuss the different reactions of plants to light. There are two main light sensing systems in plants. That is phytochrome sensing system, red light sensing system that affects plants flowering, seed germination, etiolation, senescence, abscission of leaves, and circadian rhythm. Then the blue light sensing system. This regulates the plant response such as phototropism, stomata opening, and the chlorophyll synthesis. Phototropism. The plant response to light is achieved by the action of the hormone auxin produced at the apical meristem, but actively transported to zones of elongation. Unidirectional illumination causes the auxin to move and accumulate on the shaded side of the shoot and promotes faster cell elongation on the shaded side to cause the shoot to grow bending towards light. Uniform illumination causes auxins to move and distribute uniformly throughout the shoot, hence promoting equal and upright growth of the shoot. Stomato opening is initiated when blue light activates the conversion of starch into malic acid and proton hydrogen ions in exchange for proton hydrogen ions inside the guard cells. Guard cells actively take up potassium ions using ATP from light reactions of photosynthesis and cytokine, a plant hormone. At the concentration of potassium ion increase in the sub-guard cells, the concentration of hydrogen ions decreases to increase the pH above 7. The sap becomes alkaline. Guard cells increase the uptake of choline, an ion, to maintain the electrical and ionic balance inside and outside. Malate ions formed in the guard cells are neutralized by the potassium ions to form potassium malate, which enter the cell sap of the guard cells to increase the solubility, solute concentration. The water potential decreases and causes the endosmosis into the guard cell, which become tagged to open the stomata. Photoperiodism. The plant response to changes in the photoperiod or relative length of daylight and darkness in each 24-hour cycle is brought about by the phytochrome sensing system, red light sensing system. During daylight, the inactive phytochrome red PRP600, which is manufactured by plants, absorbs red light and converts to phytochrome far red PFR-P730. In long day plants that only flower when the length of darkness they have been exposed to is shorter than a certain critical length of darkness, high levels of phytochrome far red stimulate the conversion of the inactive hormone precursor into fluorescein, which on transportation from leaves to buds induces flowering in long day plants. Phytochrome far red is converted back to phytochrome red slowly in the dark or rapidly if exposed to far red light, that is 730 nanometer. In short day plants that only flower when the length of darkness they have been exposed to exceeds a certain critical length of darkness, high level of phytochrome red and decreased levels of phytochrome far red stimulate the conversion of the inactive hormone precursor into fluorescein, which induces flowering in short day plants. In day neutral plants, flowering occurs irrespective of photoperiod. During germination of some seeds like lettuce, red light stimulates germination. Red light promotes the production of phytochrome far red in the seeds, which in the presence of other factors like water stimulate the synthesis of gibberellin, which stimulates the aleuron layer in the seeds to synthesize enzymes like amylase that promote germination. #56. Describe how the nerve impulse crosses a chronergic synapse. Part B. Explain the functions of the sensory neuron and a motor neuron in a reflex arc. Describe how nerve impulses cross a cholinergic synapse. When the action potential arrives at the presynaptic knob, it causes the calcium ion channels to open. The calcium ions flood into the neuron down their concentration gradient. The knob contains many tiny vesicles full of neurotransmitter acetylcholine. The calcium ions make these vesicles to move to the presynaptic membrane and fuse with it, releasing the acetylcholine into the synaptic cleft. In the cleft, or this cleft is very small, so it takes only a millisecond or two for the acetylcholine to diffuse across it. On the other side of the cleft, there are receptor molecules in the postsynaptic membrane, and the acetylcholine molecules fit perfectly in this. This makes sodium ion channels in the postsynaptic membrane open, so sodium ions flood down their concentration gradient. This depolarizes the membrane, giving it a positive charge inside, which sets up an action potential in the postsynaptic neuron. Explain the functions of a sensory neuron and a motor neuron in a reflex arc. A sensory neuron has its cell body in the ganglion in the dorsal root of the spinal nerve. It has a very long dendron that carries the action potential from the receptor towards its cell body, and a shorter auxin that carries the action potential into the spinal cord or brain. The ending of the dendron may be within specialized receptor, such as Pacinian capsules in the skin. Pressure acting on the Pacinian capsules depolarizes the membrane of the dendron and generates an action potential. The motor neuron has its cell body within the central nervous system, in the brain or the spinal cord. It has many short dendrites and a long axon. It will have many synapses, including several with sensory neurons. Thus, the action potential from the sensory neuron can cross the synapse and set up an action potential in the motor neuron, which will then transmit it to the effector, such as muscle or gland. The action potential then causes the effector to respond, for example, by contracting if it is a muscle. In reflex arc, the impulses travel directly from the sensory to the motor neuron, or sometimes via the intermediate neuron between them, without having to be processed in the brain. This means the pathway from receptor to effector is short as possible, so that the response can happen very quickly. Thank you very much for participating in coordination and control. Questions 51 to 56. Please do like, share, and subscribe.